So I was just chilling, right? Having a nice Canada day, beating the heat, having some brewskis after some steak and broccoli, and I find out motherfuckers really be out here trying to make chimeras and shit. You know what a chimera is? I'll tell you what a chimera is. They making them right here, right
Scientists
now.
at Stanford put human brain tissue into a rat. They wanted to see if the human
That shit a
neurons would merge
chimera.
with the
When you combine animal tissues that ain't supposed to combine, that's a chimera, dog.
rat's brain, enough to control its behavior and respond when they tickled its whiskers. Human cerebral organoids, or mini-brains, are grown in a lab, usually from reprogrammed skin cells, to study brain disorders. If you grow
Damn.
neurons in vitro, like a petri dish, they'll self-organize into biological neural networks with electrical communication similar to a developing brain, and maybe even some basic ability to learn.
Yeah, I heard
But
about
mini
this
brains
shit.
are hard to grow in a
They can play
lab. They
Pong.
only get up to a few millimeters wide because they don't get the blood supply or chemical signals they get in a real brain. If you want to study complex brain processes and how they go wrong and how they affect behavior, you can't model those with an in vitro organoid.
So the solution is obviously to make chimeras.
You gotta grow them in vivo, attached to a living organism. To make these mini brains more like human brains, they transplanted them into baby rats. As the rat's brain developed, its blood vessels infused the human brain tissue. They also found glial cells in the organoid, which help neurons survive and communicate in ways we don't fully understand. In this environment, with
Damn.
support from the rat brain, the transplanted organoid grew 9 times its size in 3 months. Its individual neurons got bigger than the in vitro neurons, and they were more electrically active, probably in part because they had more dendrites to pick up signals from
They
other
had more
neurons.
activity out of the way, dog.
Transplanting the organoids led to changes in their gene expression. All your cells have basically the same genes, but they use them differently. They can turn on a gene when they need the protein it codes for. If a neuron is making a new synapse, a new connection with another neuron, it might express these genes. The transplanted organoids showed more expression of a lot of genes related to neural development. Even though they were
Wow.
grown in a rat's brain, they expressed more of a
Now
gene
that's
that's
some
only
smart people
expressed in
shit.
primate brains called OSTN. Rats have a version of this gene, but it's expressed in their bones and skeletal
Wait a sec. So they really made chimeras. This shit is not shit that rat brains do.
muscles to
Goddamn.
regulate bone growth. In
We
primates,
gonna have super
OSTN
smart
was
rats
repurposed
that
to
go
regulate the
fucking
growth of
overpower
dendrites in their
AI.
increasingly complex brains.
AI
After
rats.
7 or 8 months of development, the gene expression patterns of both the in vivo and in vitro organoids was similar to a human fetus of about the same age, which does not mean they had the mind of a fetus. Brain organoids, even in vitro, seem to naturally go through a 9-month sequence of gene expression similar to prenatal human brains. The
God
transplanted
damn.
organoids look just a little more mature in terms of that progression. They're still a long way from real brains. They don't show the typical layers of human cortex, for example. But the
Of
goal is
course.
not to build a perfect
Hopefully.
human brain replica, it's to build models for studying brain disorders. The researchers built an organoid model of a rare and deadly genetic disorder called Timothy syndrome. They grew the organoids using cells from Timothy syndrome patients and
What's
compared them with
Timothy
organoids
syndrome?
grown from healthy participants, both in
Timothy.
vivo and in vitro. They found an unusual pattern of dendrite branching in the Timothy syndrome organoids that they could only see.
Man, we gonna have to find out what Timothy syndrome is. Maybe a future reaction. This stuff is smart, man. It's over my head. But I like doing that sometimes. Hopefully you do too. That's probably why you visit this channel.
In vivo in the
Subscribe, please.
transplanted organoids. So these in vivo organoid models might reveal things about brain disorders that we wouldn't see with these dollar store in vitro organoids. Another thing you can do with in vivo models is look at how different brain areas talk to each other to make things like perception and behavior possible. If the organoid can interact with the right brain networks, we can look at the effects of new treatments on the organism as a whole instead of just brain cells in a dish. So how much did these human cells merge with the rat's brain? When the rat pup was 3 to 7 days old, they put the mini
Rat
brain on
pups.
its somatosensory cortex, S1. S1 processes sensory input from the rat's body, or soma. If you tickle a rat's whiskers, S1 lights up. Researchers wanted to know, would the organoid respond to the rat's sensations? A week or two after the transplant, they
wouldn't it?
electrically stimulated the rat's S1, and they saw an electrical response in the organoid. But it was even more integrated than that. S1 gets information about the whiskers from the thalamus, a subcortical area deep in the brain. They found cells from all the way in the thalamus had made their way inside the organoid. When they stimulated those thalamic cells, the organoid responded too.
Holy shit, the level of chimeraism, and I just coined that term, that's going on here, I didn't even expect when I clicked this video. So, these human neuronal organoids are actually integrating into the rat brains and kind of harmoniously functioning, improving the rat brains, like, naturally. What the fuck?
The
We are
whiskers
gonna have
are
super
a
smart
major
rats.
sense organ for rats. A lot of S1 is dedicated to the whiskers.
Watch, they
If you
gonna be
tickle
able
this
to
whisker,
sense
you'll get a
fucking
response there on
5G through their
S1.
whiskers now.
So if you want to activate an organoid on top of S1, the scientific approach is whisker tickles. Here's the resting activity of some human neurons in the mini brain when the whiskers are not being tickled. When the researchers touch the whiskers, this happens. So we can see that
What
the
the
human mini brain
bumbarasa?
is getting sensory input from the rat's body. That brings us to the final and critical test. Does it work the other way around? Can the organoid control the rat? We saw that the rat neurons made it inside the organoid. How far inside?
Bro, they could implant fucking organoids that are like parasites that could control their hosts. What the fuck?
The rat
They
brain
can
did
make
the
it
human
into a
neurons travel?
vaccine.
One
Oh
way
my
to
God,
find
we're
out is
going
to
deep
infect the
now.
organoid with a modified rabies virus. Then you transplant it into the rat, let it grow for a
Bregin,
couple weeks, and look for the
Bregin, we was making chimeras, now we modify rabies. Sheesh.
rabies markers in the rat's brain. They found human cells marked with rabies in the rat's S1 and in the thalamus, so the human cells were deeply integrated into the rat's brain. When they used a different virus called a lentivirus, they found human neural projections all over the rat's brain: motor cortex, auditory, prefrontal, hippocampus. They used that same lentivirus to genetically modify the organoid so it would grow photoreceptors that respond to blue light, a technique called optogenetics.
Damn.
When they shined a blue light on the organoid, they found an
Yo, this
electrical
is some
response not only
crazy
in the
smart
organoid,
shit.
but
Like,
also
who does
in the rat
MIT
cortex,
does this?
which means the organoid is plugged into the rat, sending signals to
Bro,
its
I'm gonna
brain
need to do
in
some
addition to
dumb
getting
shit
sensory
after
signals
this.
from it. But the researchers wanted to send more than an electrical signal. They wanted to use the mini brain to trigger a whole behavior, drinking water, which you definitely can't do in vitro. Here's how they trained the rats. They shined
They did not mind control rats using
a light on their
organoids.
organoid, alternating between blue and red for 5 seconds each. The blue light activated the organoid. If they licked when the blue
What
light was
the
on, they got
fuck?
water as a reward. If they licked when the red light was on, they got nothing. After a couple weeks, the rats were licking significantly more when their organoid was activated by the blue light. So the researchers activated rat neurons to drive reward-seeking behaviors,
Blue light
which you can think of
affecting
as rat mind
organoids
control,
in the
but really it's
brain.
a lot like Pavlov's dog,
The
just with
third
a ball
eye,
made of genetically
bro.
modified human brain tissue
It
plugged into
sees
its brain.
shit. I'm
That's all.
telling you.
If this organoid transplant can affect the rat's behavior,
It
how
see
else
them
could it affect them?
frequencies, bro.
The researchers looked for effects on memory, motor activity, and fear learning. The transplanted rats did fine on a memory test. In the open field test, you let the animals run around, not in an open field, but in a little box, and you see how much they run around and explore. Transplanted rats look normal. To test their fear learning, they trained the rats to be afraid of a room that smelled like mint, but the transplanted rats didn't look any more or less scared than the normal rats.
Yeah.
A previous study found that a human organoid transplanted deeper into a mouse's brain did increase the fear response, leading to more freezing when they heard a tone that signaled a shock.
What the fuck, man?
Animal research
Look at these names. So that's what they're doing in China. Xindong, Shiboju, Xinchen. God damn. Molecular psychiatry. Bro, there's a journal of molecular psychiatry. I didn't even know that was a thing, and I have a bachelor's in psychology. The fuck?
Researchers always have to weigh the benefits of their research against the potential harm to animals. A lot of this organoid research is designed to treat neurodevelopmental disorders, so the potential benefit is huge. But how do you weigh that against the possibility of creating a self-aware rat or a conscious organoid?
No.
As mini-brain research advances,
Conscious, organoid, self-aware rats. Like, we getting there, man. Rats are gonna beat out AI.
those are the questions that researchers, ethics boards, and even lawmakers will have to wrestle with.
The ethics boards always come after the discoveries, man. It's just like AI. We gonna let the cat out the bag and then decide what to do with it. I am your boy Tony Poke, the E-Boy Zombie Slayer. Ha. About to do some dumb shit. That was way smart. Play some video games or something. Watch some pole dance. Smash subscribe. Drop me a comment. Till next time.